Storage. QIAamp mini columns should be stored at 2 to 8 degrees Celsius upon arrival and are stable for at least 1 year after delivery, if not otherwise stated on the label. However, short-term storage, up to 4 weeks, at room temperature 15 to 25 degrees Celsius does not affect their performance. All buffers can be stored at room temperature for up to 1 year, if not otherwise stated on the label. Lyophilized carrier RNA can be stored at room temperature. Carrier RNA can only be dissolved in buffer AVE. Dissolved carrier RNA should be immediately added to buffer ACL, as described on Table 2, page 27. This solution should be prepared fresh and is stable at 2 to 8 cells for up to 48 H. Unused portions of carrier RNA dissolved in buffer AVE should be frozen in aliquots at 30 to -15 degrees Celsius. The QIAamp Circulating Nucleic Acid Kit contains a ready-to-use proteinase K solution, which is dissolved in a specially formulated storage buffer. The proteinase K is stable for up to 1 year after delivery when stored at room temperature, if not otherwise stated on the label. To prolong the lifetime of proteinase K, storage at 2 to 8 degrees Celsius is recommended. Intended use. The QIAamp Circulating Nucleic Acid Kit is intended for molecular biology applications. This product is not intended for the diagnosis, prevention, or treatment of a disease. All due care and attention should be exercised in the handling of the products. We recommend all users of QIAamp products to adhere to the NIH guidelines that have been developed for recombinant DNA experiments or to other applicable guidelines. QIAcube Connect is designed to perform fully automated purification of nucleic acids and proteins in molecular biology applications. The system is intended for use by professional users trained in molecular biological techniques and the operation of QIAcube Connect. Safety information. Do not add bleach or acidic solution directly to waste containing buffer ACL, buffer ACB, or buffer ACW1. Buffer ACL, buffer ACB, and buffer ACW1 contain guanidine salts, which can form highly reactive compounds when combined with bleach. If liquid containing these buffers is spilt, clean with suitable laboratory detergent and water. If the spilt liquid contains potentially infectious agents, clean the affected area first with laboratory detergent and water, and then with scent, sodium hypochlorite. Introduction. Free circulating nucleic acids, such as tumor-specific extracellular DNA fragments and mRNAs in the blood or fetal nucleic acids in maternal blood, are present in serum or plasma, usually as short fragments. In addition, free circulating miRNAs as small as 20 nt have the potential to provide biomarkers for certain cancers and disease states. The concentration of free circulating nucleic acids in biological fluids, such as plasma, serum, or urine, is usually low and varies considerably among different individuals. For free circulating DNA in plasma, the concentration can range from 1 to 100 nanomoles in human samples. In samples obtained from different individuals, a similar sample-to-sample variability can be assumed for the concentration of circulating messenger RNA fragments and miRNA molecules. The QIAamp Circulating Nucleic Acid Kit enables efficient purification of these circulating nucleic acids from human plasma, serum, or urine. Samples can be either fresh or frozen, provided that they have not been frozen and thawed more than once. Extension tubes and vacuum processing on the QIVac 24 Plus enable starting sample volumes of up to 5 milliliters and flexible elution volumes between 20 micronanoliters and 150 milliliters allow concentration of nucleic acid species that are present in low amounts in the sample material. The kit can also be used for purification and concentration of viral nucleic acids from large sample volumes. Free circulating cell-free DNA, RNA, or viral nucleic acids are eluted in buffer AV, ready for use in amplification reactions or storage at -30 to 15 degrees Celsius. Purified nucleic acids are free of proteins, nucleases, and other impurities. Principle and procedure. The QIAamp Circulating Nucleic Acid procedure comprises 4 steps. Lyse, bind, wash, elute, and is carried out using QIAamp mini columns on a vacuum manifold. The robust procedure helps to eliminate sample-to-sample cross-contamination and increases user safety when handling potentially infectious samples. The simple procedure, which is highly suited for simultaneous processing of multiple samples, provides pure nucleic acids in less than 2 H for 24 samples. If the QIAamp Circulating Nucleic Acid Kit is used for isolation of viral RNA and DNA, the performance cannot be guaranteed for every virus species and must be validated by the user, period. Sample volumes. QIAamp mini-columns can bind fragmented nucleic acids that are as short as 20 bases, but yield depends on the sample volume and the concentration of circulating nucleic acids in the sample. The QIAamp Circulating Nucleic Acid procedure has been optimized for large sample volumes of up to 5 millimeters. Lysing samples. Free circulating nucleic acids in biological fluids are usually bound to proteins or enveloped in vesicles, requiring an efficient lysis step to release nucleic acids for selective binding to the QIAamp mini-column. Hence, samples are lysed under highly denaturing conditions at elevated temperatures in the presence of proteinase K and buffer ACL, which together ensure inactivation of DNases and RNases and complete release of nucleic acids from bound proteins, lipids, and vesicles. For processing of urine samples and for extraction of microRNAs using the microRNA protocol on page 53, buffer ATL is required as additional lysis buffer to ensure complete release of nucleic acids in the sample. Buffer ATL is available separately. Adsorption to the QIAamp mini membrane. Binding conditions are adjusted by adding buffer ACB to allow optimal binding of the circulating nucleic acids to the silica membrane. Lysates are then transferred onto a QIAamp mini-column, and circulating nucleic acids are adsorbed from a large volume onto the small silica membrane as the lysate is drawn through by vacuum pressure. Salt and pH conditions ensure that proteins and other contaminants, which can inhibit PCR and other downstream enzymatic reactions, are not retained on the QIAamp mini membrane. A vacuum manifold, e.g. the QIAvac 24 Plus with the QIAvac connecting system, and a vacuum pump capable of producing a vacuum of -800 to 900 millibar, e.g. QIAGen vacuum pump, are required for the protocol. A vacuum regulator should be used for easy monitoring of vacuum pressures and convenient vacuum release. Removal of residual contaminants. Nucleic acids remain bound to the membrane, while contaminants are efficiently washed away during 3 wash steps. In a single step, highly pure circulating nucleic acids are eluted in buffer AB, equilibrated to room temperature. Elution of pure nucleic acids. Elution is performed using buffer ABE. The elution volume can be as low as 50 microliters. If higher nucleic acid concentrations are required, the elution volume can be reduced to as low as 20 microliters. Low elution volume leads to highly concentrated nucleic acid eluates. For downstream applications that require small starting volumes, e.g. some PCR and RT-PCR assays, a more concentrated eluate may increase assay sensitivity. For downstream applications that require a larger starting volume, the elution volume can be increased up to 150 microliters. However, an increase in elution volume will decrease the concentration of nucleic acids in the eluate. The eluate volume recovered can be up to 5 microliters less than the volume of elution buffer applied to the column. For example, an elution volume of 20 microliters results in more than 15 microliters final eluate. The volume of eluate recovered depends on the nature of the sample. Eluted nucleic acids are collected in 1.5 milliliter microcentrifuge tubes, provided. If the purified circulating nucleic acids are to be stored for up to 24 hours, storage at 280 Celsius is recommended. For periods of storage longer than 24 hours, storage at 30 to 15 degrees Celsius is recommended. Yield and size of nucleic acids. Yields of free circulating nucleic acids isolated from biological samples are normally below 1 microgram and are therefore difficult to determine with a spectrophotometer. In addition, carrier RNA present in the extracted nucleic acids is likely to dominate UV absorbance readings. Quantitative amplification methods are recommended for determination of yields. The absolute yield of circulating DNA and RNA obtained from a sample using the QIAamp Circulating Nucleic Acid Kit varies considerably between samples from different individuals and also depends on other factors, e.g. certain disease states. The QIAamp Circulating Nucleic Acid Kit delivers excellent recovery of fragmented nucleic acids as short as 75 BP. The recovery of microRNAs as short as 20 min is further enhanced when protocol, purification of circulating microRNA from 1 milliliter, 2 milliliters, or 3 milliliter serum, plasma, or urine is used. The size distribution of circulating nucleic acids purified using this procedure can be checked by agarose gel electrophoresis and hybridization to a target-specific labeled probe. Description of protocols. Different protocols are provided in this handbook for different volumes of starting material. Protocol: purification of circulating nucleic acids from 1 milliliter, 2 milliliter, or 3 milliliter serum or plasma is for processing more than 3 milliliter serum or plasma. Protocol: purification of circulating nucleic acids from 4 milliliter or 5 milliliter serum or plasma is for processing 4 to 5 milliliter serum or plasma. Protocol: purification of circulating nucleic acids from 1 milliliter, 2 milliliter, or 3 milliliter urine is for processing less than or equal to 3 milliliter urine. Protocol: purification of circulating nucleic acids from 4 milliliter urine is for processing up to 4 milliliter urine. In addition, protocol: purification of circulating microRNA from 1 milliliter, 2 milliliter, or 3 milliliter serum, plasma, or urine is designed for purification of circulating miRNA, including total circulating RNA from 3 milliliters of plasma, serum, or urine.